Good evening. Our group will be presenting on cellular response to stress, cellular responses to nutrient deprivation. Our group members include Lizu Prasad, Shamia Conway, Felicia Mohammed, Alex Loy, Krishna Jinarain, Arella Williams, Tramil Daniels, and myself, Sharifa Khan. Contents will be covering 1. The introduction, 2. Molecular responses to nutrient deprivation in cells, 3. Role of stress response proteins in cellular damage mitigation, 4. Exploring signaling pathways for mitigating cellular damage, and lastly, our conclusion. The introduction. Cells activate a stress response involving various proteins to address nutrient deprivation. These proteins mitigate the effects of nutrient stress, preserve cellular structures, and maintain energy balance and counter oxidative damage. These adaptation mechanisms help cell conserve resources and overcome challenges during scarcity. Now, before we go into the presentation, we need to know exactly what nutrient deprivation really is. Now, the definition. Nutrient deprivation refers to a condition where cells, tissues, or organisms experience an inadequate supply of essential substances required for normal physiological functions, or more specifically, engage in specific actions. Now, the importance. Nutrients are crucial substances that organisms need for growth, maintenance, and proper functioning at the cellular level. Molecular responses to nutrient deprivation in cells. 1. When the body needs nutrients, pathways are activated which signal cells to become more active, like a superhero power. 2. AMPK, activated protein kinase, plays a crucial role in regulating energy levels in the body, particularly in response to low nutrient levels. 3. AMPK plays a vital role in maintaining cellular function during nutrient deficiency, making it a significant factor in cell biology. I'll now hand you over to the next presenter.
Lisa, go back, please. Thank you. Yes, good evening. 4. When food is limited, the body stops anabolic pathways that aid growth and focuses on conserving energy, which is located in the body, as we know. Halt. In order to survive when food is scarce, cells know how to make some tough decisions. They cut back on protein, making acellular growth, wisely allocating precious energy resources to keep the lights on. Basically, they know how to conserve and use less energy within the body so they may be able to survive. 6. Efficient energy usage, a lesson from cells. When resources are limited and times are tough, cells know how to conserve energy. They focus on the essential and minimize the non-essential. Basically, they focus on the specific energy they need to use to function, and use less of the energy they don't need. Now I'll hand you over to the next presenter.
Good evening once more. I will be presenting on the role of stress response proteins in cellular damage mitigation. Now, when cells lack nutrients, they are at risk of dying. In response, they activate a sophisticated stress response that involves specialized proteins acting as guardians, safeguarding the cells and promoting their survival. And here we have 4 types of stress response proteins. There is the heat shock proteins, the unfolded protein response, the autophagy proteins, and the RNA chaperone proteins. I'll now hand you over to Tramel.
Preventing proteins functions. HSPs and UPR proteins prevent protein misfolding and aggregation, protecting vital cellular process, maintaining cellular energy balance. Autophagy recycles cellular components and provide essential nutrients for energy production, even under desperation. Converting resources. Increase proteasomes activities and autophagy ensures essential. Oh my gosh, sorry. Hold on 1 minute, no. Sorry about that. Utilization of limited nutrients and maximizing cell survival. Next slide. Felicia, use next there.
Yes. So moving on, guardians of cellular integrity, stress response proteins. Now we're going to talk a bit about the heat shock proteins and the chaperone proteins. Now the heat shock proteins, or the HSPs, are like superheroes that keep our cells in top shape when our bodies are running on empty. They step in during nutrient shortages to protect against protein structures and stop them from clumping together. HSPs work tirelessly to make sure those proteins are folded correctly and ready to do their job. And the chaperone proteins, when the cells are running low on nutrients, chaperone proteins swoop in like superheroes, ensuring that protein folding is done with maximum precision, reducing the risk of any errors. They also double up as vigilant watchmen, making sure that the proteins meet the strict criteria for flawless cellular function. I'll now hand you back to Tramel Daniels.
Okay. Autophagy, a cellular cleanup crew in nutrition structure. Scarcity, sorry about that. Picture a tiny recycling plant operating 24/7, keeping our cells in top shape. Autophagy is an ultimate superhero of cellular process, working to keep things running smoothly. When cells face a famine or nutrient shortage, autophagy jumps into action, prioritizing important stuff and getting rid of the rest. But that's not all. Autophagy also helps clear out any damaged organelles or proteins, keeping our cells healthy and happy. Okay, next person.
Good evening. I'll be taking you through the cell signaling pathways. Exploring signaling pathways for mitigating cellular damage. Now, cell signaling pathways are how cells communicate with each other to carry out their functions. Exposure to stress disturbs cellular homeostasis, and as a result, initiates signaling pathways that help the cell's metabolic processes return to balance. Now, one example of such pathway is the unfolded protein response, the UPR, of the endoplasmic reticulum, ER. The unfolded protein response is activated during endoplasmic reticulum stress, which is triggered by an accumulation of unfolded proteins. It helps cells cope by reducing protein synthesis and increasing ER folding capacity. Now, under normal circumstances, the UPR is not triggered, but it can become engaged in response to specific stresses when there's an accumulation of unfolded proteins, as mentioned previously. So in response to this, the UPR acts to help mitigate cell stress by clearing those unfolded or misfolding proteins and increasing the capacity of the ER to then fold those proteins. Another example of such pathway is the CJON N-terminal kinase, the JNK, or the P38 MAPK, the mitogen activated protein kinase signaling pathways. Now, these pathways respond to stress, like metabolic stress, from a high-fat diet. Prolonged activation is linked to insulin sensitivity changes and metabolic issues in obesity, influencing gene regulation and cellular processes. Now, these signaling pathways regulate the body's reaction to both internal and external stressors. So in response to such cell stress, these pathways regulate gene expression, mitosis, metabolism, motility, and cell survival. And by doing so, they reduce or mitigate cell stress. I'm going to hand over to the next presenter.
Next slide, please. So during nutrient deficient induced stress, cellular responses are orchestrated through pivotal signaling pathways. So the mTOR pathway is known for its sensitivity to nutrient availability, and it regulates cell growth, metabolism, and autophagy. When nutrients are scarce, the mTOR activity decreases, triggering autophagy. When recycles, recycles cellular components to maintain energy level and support cell survival. Simultaneously, the AMP, activated protein kinase pathway, activated by an increase of the AMP and ATP ratio in response to nutrients limitation, inhibits anabolic processes and promotes catabolic pathways. This helps to restore cellular energy balance, and these pathways collectively contribute to cellular adaptation, energy homeostasis, and survival during periods of energy deficiency-induced stress. Over to the next person.
Now here we have an image illustrating the stress signaling pathways activated in response to metabolic stress. So at the top, we can see there is illustrated a high-fat diet. Now, individuals feeding on a high-fat diet all the time may be deprived of nutrients, which we need for growth. And as a result, it may cause metabolic stress, as we can see there. Now, this metabolic stress, as a result, can lead to the activation of the UPR and the MAPK stress-activated proteins. Now, activation of these signaling pathways then results in adaptive response associated with obesity, as we can see there, and altered insulin sensitivity, which helps them mitigate cell damage. So as the body goes through metabolic stress, the signaling pathways respond, and as a result, they help to mitigate the cell damage, or they help to reduce the cell from dying. Next slide, please. In conclusion, the web of responses to nutrient deprivation underscores the vital role stress response proteins play in safeguarding cells. From detecting nutrient stress to coordinating complex signaling pathways, these proteins aid in mitigating damage and promoting cell survival. The adaptive strategies employed by cells, such as conserving resources and activating specific pathways like UPR, the JNK or P38 MAPK, the mTOR and AMPK signaling pathways demonstrate their resilience in the face of nutrient challenges. So in this presentation, we basically covered ways in which the pathways and proteins can help mitigate cell damage as a response to stress. The next slides we have our references, and that brings us to the end of our presentation. If you have any questions, please feel free to reach out to any of the group members. Thank you.